Hello everyone, my name is Aj Keefe, and in this presentation I'll be covering the paper Ubiquitin C-terminal hydrolase L1, UCH-L1, promotes hippocampal memory via its deubiquitinating effect on TrkB, published in the Journal of Neuroscience. I want to extend a special thanks to the authors for their contribution to this field of research, and I recommend that my viewers pause the video at each new slide and read through the material and analyze the graphics before playing my commentary. And if you have any questions, please comment below. I hope you enjoy. In this paper, we'll be discussing the modulation of the TrkB or TrkB receptor. So let's review the BDNF TrkB signaling pathway. So BDNF, which stands for brain-derived neurotrophic factor, is an incredibly important protein that is trafficked to synapses, released, and then very effectively supports the survival of the target neuron. So before jumping into specifics, let's first think about what it means for one neuron to support the survival of its synaptic partner. So some neuronal populations of the brain, most notably the striatum and the hippocampus, appear to be uniquely reliant on an adequate supply of BDNF. A loss of BDNF support from other neuronal populations, such as from the frontal cortex innervating the striatum, sensitizes the striatal cells to the kinds of degeneration we see in Huntington's disease. A loss of BDNF innervation is also implicated in Alzheimer's disease and degeneration of hippocampal neurons. And this is an interesting phenomenon because dysfunctions in BDNF transcription, translation, and trafficking inside of entirely different neuronal populations can fail to promote the survival of another set of neurons. In other words, it's possible in diseases such as Alzheimer's or Huntington's disease that dysfunction begins not in the hippocampus or the striatum, but rather in the neurons that are supposed to be supporting their survival through BDNF. For example, in Huntington's disease, a major avenue of research is into whether dysfunctional cortical neurons are failing to deliver BDNF into striatal cells, and that's why they selectively die. So it's easy to become hyper-focused on the cells that die in any particular disease. But since the brain is so interconnected, it's possible that the pathology could begin in entirely different neuronal populations. Another more general reason to be interested in BDNF comes from simple studies of what happens when BDNF is knocked out or upregulated. And the homozygotic deletion of BDNF causes embryonic lethality, but conditional deletion in mice after they've already developed and everything causes enhanced susceptibility to neurodegeneration, also causes impaired learning, impaired hippocampal neurogenesis, and many other problems that are mostly confined to the brain. And the overexpression of BDNF basically does the opposite, where it significantly protects against neurodegeneration, it enhances memory, and it enhances neurogenesis. So I hope by this point I've convinced you more broadly that BDNF is just a critical signaling pathway, and so understanding how it works is definitely a worthwhile endeavor. So how does BDNF actually promote cell survival? Well, cleaved BDNF binds to TrkB receptors. So in this graphic, we have NGF, nerve growth factor, binding TrkA. BDNF binds to TrkB, very similar interaction going on. So cleaved BDNF, which I mentioned cleaved specifically because I don't want to digress too much, but non-cleaved, unprocessed BDNF is actually a potent pro-apoptotic signaling protein. So completely diametric outcomes based on whether BDNF has been cleaved or not. So the processing and cleavage of BDNF is very critical, but that's a different story. I won't get into it here. But after its release, BDNF binds to TrkB, a receptor tyrosine kinase. So when BDNF binds to TrkB, or TrkB, it causes membrane-bound TrkB receptors to dimerize and transphosphorylate each other. And so all BDNF actually does is that it promotes cell survival by inducing the dimerization of TrkB receptors. And when this tyrosine kinase TrkB dimerizes, it forms a powerful pro-survival signaling platform. And the three main mechanisms by which TrkB promotes survival are through the AKT, the ERK, and the calcium calmodulin signaling pathways. We see in this graphic, we have ERK signaling. We have CrEB. CrEB is the calcium activated. We also see over here we have ERK, and we're missing AKT, but AKT is very important. So AKT is actually probably the most important signaling molecule because loss of AKT alone can cause axon degeneration and cell death. And it promotes survival by negatively regulating or inhibiting the JNK and the GSK3 beta stress signaling pathways. So AKT inhibits stress signaling, and AKT also phosphorylates and inhibits the BH3 protein, BAD, which then, or when it's dephosphorylated, it can actually promote cell death by forming holes in the mitochondria. So AKT constitutively inhibits BAD. AKT can also directly inhibit caspase-9 and thus directly inhibit apoptosis by phosphorylation. And AKT can also activate mTORC to upregulate translation and promote memory formation. So AKT is a very critical signaling protein for neurons, and much of it stems from much of the AKT signaling in a neuron comes from BDNF. Another really important player is ERK. So ERK. I'm not very familiar with this protein, but it essentially just leads to pro-survival transcription through RSK. It's a transcription factor. BDNF also activates the classic calcium calmodulin pathway that upregulates cytosolic calcium concentrations to activate the phosphorylation of the CrEB transcription factor. And CrEB is basically synonymous with long-term potentiation or memory, and the loss of CrEB is integral to the pathology of multiple neurodegenerative diseases. So BDNF activation of TrkB promotes cell survival through AKT, ERK, and calcium calmodulin activating CrEB, and probably many more mechanisms as well. So TrkB dimerization induced by BDNF is just critical for a neuron's survival. So what else is happening at the synapse besides TrkB dimerization and transphosphorylation? Well, there's many things happening, but one critical event that's often underappreciated is that TrkB activation is often followed by internalization of the receptor by endocytosis. So monoubiquitination, which is a well-understood flag of receptor endocytosis and sorting, is also critical for TrkB endocytosis. When TrkB is monoubiquitinated, which it usually occurs after BDNF binds to TrkB, it recruits the endocytic machinery and it's internalized. We see this happening with TrkA as well. It's been very well studied in motor neurons. And when TrkB is internalized by this monoubiquitination, it is retrogradely transported in this vesicle towards to the nucleus or the cell body. And this has the effect of moving that potent pro-survival signaling platform closer to the nucleus where transcription can promote long-term changes in cellular well-being. We see out here in the synapse that we still have ERK signaling in PI3K, but this is still, it could be a very long way from the cell body. So when this vesicle is retrogradely transported, it's amplifying these pro-growth, pro-survival signals right next to the nucleus to influence gene expression. So in order for a cell to get the whole package, TrkB receptors are retrogradely trafficked towards the nucleus. Monoubiquitination, however, which causes the endocytosis of the vesicle, is only a couple of residues away from polyubiquitination, which instead of promoting endosomal trafficking and sorting, promotes proteasomal degradation. So the difference between these two outcomes couldn't be more important. I mean, just a couple of ubiquitin ligations lie between pro-survival endosome trafficking and TrkB degradation. And that brings me to this next related topic, and that is deubiquitination. So there are specific deubiquitinases that the researchers are interested in this paper, and the specific deubiquitinase that they're interested in is UCH-L1, because UCH-L1, first of all, it's highly expressed in the brain. It has already been strongly implicated in promoting memory formation, and it's also known to stabilize monoubiquitination modifications. So by cleaving and degrading polyubiquitination, UCH-L1 promotes monoubiquitination, which might sound somewhat paradoxical, but it's almost as if UCH1 will cleave ubiquitin until it becomes monoubiquitinated, and then it'll leave it alone. And so it may even stabilize these monoubiquitinated substrates, such as TrkB. And furthermore, the loss of UCH-L1 causes a profound age-dependent brain degeneration phenotype. But overall, though, the mechanism by which UCH-L1 promotes cell survival is not very well understood. And so the authors of this current paper wondered if UCH-L1's deubiquitination ability and monoubiquitination stabilization has anything to do with TrkB signaling and possibly trafficking. And so that's what we're going to be investigating in this paper. The impetus for this research was based on the idea that both TrkB and UCH-L1 are both critical for memory formation and neuronal survival. And since TrkB signaling is drastically enhanced by monoubiquitination-induced endosomal trafficking, and also by the fact that UCH-L1 promotes monoubiquitination, perhaps both these proteins are coordinating in some way to promote TrkB's pro-survival signaling. And it turns out that this is indeed the case, and this is specifically what they discovered. First, they observed that short hairpin-mediated knockdown of TrkB or UCH-L1 directly into the hippocampus impairs memory, which is substantiated by previous literature. They also found that these hippocampi that had been transduced with shRNA towards UCH-L1 had increased TrkB ubiquitination and decreased TrkB expression. And overexpression of UCH-L1 also using viral transduction increased TrkB stability and decreased TrkB ubiquitination. So these initial sets of experiments suggest that the deubiquitinase UCH-L1 is modulating TrkB expression by degrading its ubiquitinations. And these first initial experiments were all done in vivo with mice. Next, the authors did some co-immunoprecipitations, and they found a clear interaction between TrkB and UCH-L1 that was upregulated following BDNF treatment. They also did a bunch of very thorough and classic molecular biology to discover exactly which residues were interacting between UCH-L1 and TrkB1. And I won't get into that part of the paper, but suffice it to say, they discovered that residues 75 to 85 of UCH-L1 were binding to TrkB. So knowing that these 10 amino acids have a very strong affinity for TrkB exactly at the interface where TrkB binds to UCH-L1, they developed this very fascinating construct that could prevent TrkB UCH-L1 interactions. So they took these 10 amino acids that bind to TrkB at the interface, and they fused it to a protein called TAT, T-A-T. TAT is a protein derived from the HIV virus and is responsible for permeabilizing the membrane for viral entry. So by fusing TAT to this short 10 amino acid fragment, it would essentially allow the peptide to enter cells, because normally a 10 amino acid peptide is far too large to diffuse into a cell. So this UCH-L1 75 to 85 peptide fused to TAT would essentially be, it could be added to cells and it would infect them as well as a typical virus would. And then once this fragment is delivered, it would competitively block endogenous UCH-L1 from binding to TrkB. So that they have developed this very specific inhibitor of the UCH-L1 TrkB interaction. And the effect of selectively blocking this TrkB's interaction with UCH-L1 result in a couple of very interesting deficiencies. UCH-L1 7585 TAT created cells had increased TrkB internalization, which is dependent on ubiquitination. They also had decreased recycling of TrkB to the membrane, and instead TrkB was being degraded by the proteasome. So when the interaction between UCH-L1 and TrkB is inhibited, it decreased recycling of TrkB and resulted in its degradation. It also had the effect of ultimately reducing AKT and ERK signaling and reducing the pro-survival signals that TrkB would normally be emitting. They extended these findings to, so this was all done in vivo, in culture, what I just described. They also extended these findings to the in vivo live animal by stereotaxically injecting these infectious UCH-L1 7585 TAT proteins directly into the dentate gyrus of live animals. So they're injecting this little TAT-fused peptide that will inhibit the interaction. And the introduction of this peptide resulted in decreased performance in a memory-related task. And later, when these animals were sacrificed and their dentate gyrus was extracted, it showed significantly less TrkB expression following these memory tasks compared to controls. So it's a very fascinating paper, and I'm excited to get into the specifics. Their first step was to transduce, so they're using a virus to introduce all these different various constructs that you see here into the dentate gyrus of live mice and assessing their behavior on a memory task. So this percent time freezing, I'm not totally familiar with the memory task, but essentially the higher this is, the better their memory. And so what they did is they would inject these viruses, they would wait 15 days to allow the inflammation to settle down and expression to normalize. And then they did this behavioral task. They got some data, and then they sacrificed those animals and took a look at what was going on in those neurons. So on the right, we see the results of this behavioral memory task and the knockdown of TrkB, not surprisingly, hindered memory formation. In contrast, the upregulation of UCH-L1, so increasing the amount of UCH-L1 wild type, enhanced memory. And an enzyme-dead version of UCH-L1, which lacks its deubiquitinase ability, didn't do anything to its memory, which makes sense again, because endogenous UCH-L1 is still functioning. So on the bottom are the results of a Western blot conducted to confirm that TrkB was successfully knocking down TrkB. So in this condition, we see TrkB shRNA towards TrkB was successfully knocking down TrkB in the dentate gyrus, but not in the other hippocampus or the cortex or the striatum. So we know it's specific for the dentate gyrus. We also see overexpression of UCH-L1 overexpressing UCH-L1 in the dentate gyrus and nowhere else. So UCH-L1 overexpression enhances memory, and TrkB knockdown disrupts memory. So these same animals that went through those behavioral tasks had their brains, unfortunately for them, removed, and the amount of TrkB ubiquitination and expression was assayed by Western blot analysis. When UCH-L1 was overexpressed, we're seeing with this OE condition, OE, when UCH-L1 was overexpressed, we see more TrkB, and we see decreased ubiquitination of TrkB relative to controls. To confirm that UCH-L1 was directly ubiquitinating TrkB, they did a couple of co-immunoprecipitations in these easy to work with HEK or HEC 293 cells. So on the right, we see that when when UCH-L1 is knocked down, so we're 1, 2, and 3, these conditions are siRNA towards UCH-L1, and then the cells are treated with BDNF, that TrkB co-immunoprecipitates with ubiquitin. So they are pulling down, the IP is TrkB, so they're pulling down TrkB, and then they're staining for ubiquitin. And ubiquitin only shows up in the lanes that had been previously treated with BDNF. So at 15 minutes after BDNF treatment, we see ubiquitin TrkB pull down. And the ubiquitination of TrkB is potentiated by siRNA-mediated knockdown of UCH-L1. We see in the scramble, the normal control condition with no shRNA or scrambled shRNA, we see a slight co-immunoprecipitation, but is potentiated when UCH-L1 is knocked down. So in other words, BDNF promotes TrkB ubiquitination, perhaps to induce endocytosis, and UCH-L1 functions to downregulate TrkB ubiquitination. So on the bottom, we have a highly specific co-IP because they're using fusion tags, which can be purified or stained for with perfection. So attached to MYC or Mike, attached to Mike is UCH-L1. So they are immunoprecipitating or pulling down UCH-L1. And attached to TrkB is flag. So they are staining for TrkB by staining for flag. And when this co-IP is conducted, we see a significant interaction between these two proteins right here. And on the bottom right is a more conventional co-IP, simply using antibodies towards the two proteins. And again, we see that co-immunoprecipitation of UCH or of TrkB, so they're immunoprecipitating TrkB, and then they're staining for UCH-L1. And we see that this band right here indicates a interaction because they are pulling down together. So I skipped a fairly large portion of this paper that was dealing with the identification of the residues interacting between UCH-L1 and TrkB. That was done through the additional co-immunoprecipitations. And then they also further verified this TAT UCH-L1 7585 protein that they created. And they found that it was successfully inhibiting endogenous UCH-L1 TrkB interaction. So I'll just quickly summarize it for you. They found that TAT UCH-L1 7585 was very potently binding TrkB and preventing UCH-L1 from deubiquitinating it. So whenever you see TAT UCH-L1 7585, which we'll see plenty of times, whenever you see that, it means that the UCH-L1 TrkB interaction is being inhibited. When this interaction is inhibited in primary hippocampal neurons, which is what we're looking at here, we see increased internalization of TrkB, shown in red. So TrkB is in red, and the surface TrkB is in green. So to measure TrkB recycling, they did a biotin internalization assay, which briefly involves pulsing the cells with biotin and then treating them with BDNF and then staining for biotin. And so any biotin staining indicates TrkB that was previously membrane bound during the time of the initial pulse. And when TrkB UCH-L1 interaction is inhibited, we see significantly reduced recycling of TrkB, and instead it was being degraded. So in this normal control condition, we see a vehicle, so we see robust surface expression. Following BDNF treatment, we see internalization. And then when this interaction is inhibited, we see no recycling back to the surface because the UCH-L1 cannot interact with TrkB, and so there's much less of it at the surface, as seen in this picture. And on the bottom left, you see that there's more internalized TrkB in the UCH-L1 75-85 condition. This is a quantification of the previous slide's immunohistochemistry. So percent of internalized TrkB increases by about 20% when UCH-L1 TrkB interaction is inhibited. And also the amount of recycled TrkB is reduced by about 20% as well. So TrkB is being internalized and not returned to the cell surface when the interaction between UCH-L1 and TrkB is inhibited. So what effect does increased internalization and reduced recycling of the TrkB receptor actually have? Well, to test this, they measured by Western blot the amount of phosphorylated TrkB, the amount of phosphorylated AKT, and the amount of phosphorylated ERK, all downstream of the BDNF TrkB signaling pathway. And we see that following BDNF treatment in control cells, here on the left, also treated with a control TAT protein, that all these proteins are phosphorylated and are upregulated. We see plenty of phosphorylated AKT, phosphorylated TrkB, and phosphorylated ERK in 60 minutes. However, when UCH-L1 TrkB interaction is inhibited in these lanes, we see significantly less AKT right here, less phosphorylated TrkB, and less ERK signaling. Interestingly, though, we actually see perhaps a slight burst in AKT signaling, and it's theorized that this results from increased ubiquitination and mass internalization. But before degradation, this actually promotes a transient upregulation of AKT. But nevertheless, we see decreased signaling in the long term. And on the left, you see the amount of phosphorylated TrkB to non-phosphorylated TrkB is significantly downregulated when the interaction with UCH-L1 is inhibited. These couple of figures represent the coup de grace of this paper, where they are looking at TrkB expression right here and TrkB ubiquitination right here in what were previously live animals following a fear conditioning and memory formation task indicated by the CFC. So in control animals, the white bars, following a memory task. So following a memory task, we see slightly increased TrkB ubiquitination right here. We see slightly decreased TrkB protein expression, and overall we see very good memory. So they have good memory, and TrkB expression slightly drops, as well as increased TrkB ubiquitination. However, when TAT UCH-L1 75-85 is injected, only after a memory task we see increased TrkB ubiquitination relative to controls. So we see ubiquitination almost doubles when its interaction with UCH-L1 is inhibited. We also see TrkB expression drops as well compared to controls following a memory task. Without a memory task, there's no difference. Without a memory task, nothing interesting is happening. But after a memory task, we see TrkB ubiquitination is upregulated and TrkB protein is downregulated. We also see, and this is the most amazing part of this paper, that memory is significantly perturbed in these animals. So, I mean, the introduction of this very small 10 amino acid peptide into a single region of the brain, the dentate gyrus, dramatically modulates their behavior. And again, so I'm not really familiar with this behavioral task, but the comparison to controls is a very vivid difference. And this indicates that the interaction between TrkB and UCH-L1 has huge ramifications for behavior and memory, and it may even have big implications for neurodegeneration as well. So just the summary, For this paper, we have TrkB and UCH-L1 interact following BDNF treatment, and loss of UCH-L1 increases TrkB entrance into the ubiquitin proteasome system. Inhibiting the TrkB-UCH-L1 interaction significantly impairs TrkB signaling in trafficking following BDNF treatment. shRNA-mediated knockdown of UCH-L1 or selective inhibition of TrkB-UCH-L1 interaction significantly impairs hippocampal memory formation. Collectively, these results suggest UCH-L1 is critical for TrkB signaling by preventing its degradation and promoting recycling to the membrane.